Thank you.
We're going to move on to our second speaker of the day, who has been with us once before, Associate Professor James Ussher. And while James is getting his presentation set up, I'll just remind you that he's an immunologist and clinical microbiologist in the Department of Microbiology and Immunology at the University of Otago and at Southern Community Laboratories. His research interests are in applied and molecular immunology, medical microbiology, and vaccines. Today, James is going to talk about how is a COVID-19 vaccine constructed and evaluated. James.
Great. Thanks for the introduction, Jackie, and thanks again for the opportunity to come and talk to you. And thank you for joining. So today I'm going to focus on, take a global view on vaccines for SARS coronavirus 2 and try and provide an update to where things are at and what some of the challenges might be. So, sorry, let's stop sharing. I'm not sure. Miguel has already shown this slide, but just to make the point that this is something that has emerged in December last year, and from there we've gone to over 10.5 million cases and over 500,000 deaths with virus spreading around the world. So we really need vaccines to try and control the spread of this virus. And this virus isn't going to be going anywhere now. It's it's going to be with humanity for many years. So we need to find mechanisms, antivirals and vaccines to try and manage it. So just to highlight the need for a vaccine, one of the worst hit countries early on was Spain. And they conducted a Sero survey in early May, so they looked for presence of antibodies in the population to try and look at what percentage of the population might have been infected, and they found that overall about 5% of the population had been infected. At that same point, that had over 200,000 cases diagnosed and over 18,000 deaths. So you can imagine that if you're going to multiply that by 20 to get through to the whole population being infected, that's going to be cause a lot of heartache and a lot of morbidity and a lot of mortality. Similarly, Sweden has taken a different approach to many countries around the world, and that they have opted to try and slow the spread of the virus but achieve herd immunity. In a Sero survey that was published in the media back in at the end of April, at the point at which they'd had almost 3,000 deaths and over 20,000 cases, suggested that in Stockholm, where there was the highest rates of infection, they still had only 7.3% of the population showed evidence that they'd been infected. So again, there's a long way to go to achieve herd immunity, where we estimate you may need, depending on the on how effective the vaccine is, or how effective the immune response is at preventing infection, needing over 60 or 70% of the population to be immune. So now Sweden has got almost 70,000, as of today, Sweden's got almost 70,000 cases and over 5,000 deaths. And if we look at the death rate normalised to population around the globe at the moment, based on the diagnosed cases, the UK has got the highest rate of mortality rate with 648 deaths per million population. And just to put that in perspective, that equates to one person and just over 1,500 in the entire population who's died to this point from SARS-CoV-2. Similarly, New York has got a phenomenal rate of infection, and in New York that equates to a phenomenal mortality from this virus, and that equates to 1 in 262 people in New York has died from SARS coronavirus 2. So this is a major issue, and it's not going away. So a key question when we're thinking about vaccine development is whether the immune response protects against infection. And there's been a lot of debate in the Twitter sphere and in the media as to whether people have been able to be reinfected with SARS-CoV-2. the moment, there's no convincing reports that anyone has been reinfected. This report came out in Science where they looked at a monkey model. So they infected rhesus macaques with SARS-CoV-2 and then re-challenged them. So after the original infection, those monkeys developed neutralizing antibodies against the virus. They also developed cellular immunity, so they were able to detect both CD4 T helper cells and CD8 cytotoxic T killer cells that were specific for the virus. So when they initially, they then re-challenged them 35 days after the original infection. So with primary challenge, this is a log scale. So we've got the viral load over here, and we can see that the monkeys were slow to clear infection. So this is with a nasal swab and looking at the amount of virus. Whereas on re-challenge, there was very little virus detected in the nasopharynx. And that's summed up here. So just infection was minimal on re-challenge. Most of the monkeys were protected. And if we look at the immune response on re-challenge, we saw the researchers saw a rise in the antibodies, a rise in neutralizing antibodies, again log scales, and a rise in cellular immune responses against the virus. So this suggests that infection does protect against, does provide an immune response that protects against reinfection at least at 35 days post-infection. However, the question remains how durable this protection will be. So I presented this slide in the previous talk when talking about serology, but we can detect antibody responses both in asymptomatic and symptomatic patients. This is some data that was published in Nature Medicine by a Chinese group. But you see a drop in those antibody titers between acute sample, which was taken 3 to 4 weeks post-exposure, and a convalescent sample that was taken 8 weeks after discharge from hospital. So we also see a drop in neutralizing antibody titers in these patients, although they do remain detectable, and this is just summarized over here. So in the acute phase, over 80% of patients had detectable antibody responses, but that had dropped, at least in the asymptomatic, had dropped to 60% by 8 weeks after discharge. And while it remained higher, the levels had dropped in the symptomatic patients. This has been reported before, so with the original SARS coronavirus, which has now been named SARS coronavirus 1. So there was a marked decline in antibody titers after infection with SARS coronavirus 1. So this is a publication from 2007, so 4 years after the epidemic or the outbreak with SARS-CoV-1. And again, we can see in blue the percentage that positive antibody drops with time, including for neutralizing antibody. And if we look at the absolute levels, and this is on a log scale for the absolute levels, we see a marked decline in antibody levels over time. So whether this is still sufficient to provide protection against reinfection with SARS-CoV-1 is unknown because SARS-CoV-1 has stopped circulating, and so it's unknown whether that is protective. There is further data from the endemic human coronaviruses. So this is a challenge study that was performed back in 1990. So they looked here at, they had 15 volunteers who were inoculated with this human coronavirus. So the endemic human coronaviruses are responsible for between 10 and 30% of colds. And they were able to, pointer on. So of the 15 volunteers who were inoculated intranasally, 10 became infected, 8 of those became symptomatic, developing colds. And those who became infected had, this is looking at the amount of IgA or antibody that's present at mucosal surfaces and nasal washings, and the amount of IgG that was present in the serum. And those who became infected are in the black dots, whereas those who didn't are in the open dots. And you can see that those who became infected had lower levels of antibody, and that it got boosted upon infection, but that with both the uninfected and infected, it dropped off with time. So when they re-challenged them a year later, all of those who had previously been uninfected became infected, although only one developed a cold. And 6 of the 9 who had been reinfected a year ago, became reinfected, suggesting that the duration of protection, at least against infection, if perhaps not symptoms, is short. And the duration of shedding was lower after re-challenge. But that was with a homologous re-challenge. There's another challenge study where they used a heterologous virus, so from a different isolate, and a greater proportion of people who are re-challenged became symptomatic. And this preprint just came out within the last week. So they looked at over 30 years. This is a heroic study. So they looked over 30 years and 10 volunteers, and they took blood on a regular basis, and they looked for rises in antibody that would suggest reinfection. And they could show frequent reinfection with the 4 endemic coronaviruses, with a median time between infections overall of 30 months, and many infections occurring within 6 to 12 months of the original infection. So I think this raises serious concerns around, certainly in those who perhaps have been asymptomatically infected or have had a mild infection, how long their immunity may last to SARS-CoV-2. So there, as far as vaccines for coronaviruses go, most of the work has been done on the previous zoonotic coronaviruses, so SARS-CoV-1 and MERS. So the original papers on SARS-CoV-1, there was a flurry of vaccine development, and 2 vaccines made it through to phase 1 clinical trials, by which time the virus had gone away. So the efficacy of those vaccines was never able to be tested, and there was a challenging path to vaccine development, and there seemed to be less of a desire to continue developing vaccines because the virus had gone away. So those vaccine development programs were shelved. But one of the phase 1 trials was with an inactivated viral vaccine, and it was by Sinovac, which is a company's name that we'll come back to. And they showed that they could induce neutralizing antibodies with 2 injections of that virus, of that inactivated virus. Not at massive levels of neutralizing antibody, but they were able to induce neutralizing antibody. The other vaccine that was made it to phase 1 clinical trials was a DNA vaccine. So this is essentially usually a plasmid from bacteria that encodes a gene for the spike protein of the SARS coronavirus, and that gets electroporated. So basically, they use a special gene gun to try and get it into the cells in the muscle, and then it produces the antigen and stimulates an immune response. And again, we can see the different. These are different subjects. So there wasn't a great response. Some people didn't respond at all. Others had an increase in antibody levels, not to massive levels, which then declined to the orange bars. So it was able to stimulate neutralizing antibodies, but they didn't seem to persist, and they weren't of huge titer. There's also been this year 3 publications on another coronavirus, in this case the MERS virus. So this is a virus that emerged in about 2012 from memory in the Middle East, and it seems to be passed from camels into humans on a sporadic basis. So there's been 3 different vaccines that have made it through to phase 1 trials, and one of them is another DNA vaccine. And again, we can see this is looking at neutralizing antibodies as a phase 1 trial with a prime and a boost a month apart, and I'd have to say pretty disappointing levels of neutralizing antibodies stimulated by that DNA vaccine. There's also a modified vaccinia virus. So there's a viral vector that's encoding the spike protein in much the same way as the DNA viruses, vaccines do. And again, this probably stimulated some better immune responses, but still only getting up to titers of about 100. And they then started declining. So again, able to stimulate antibodies, but not massive levels. And then of interest, there's a publication from the Oxford group who have developed, developing a SARS-CoV-2 vaccine. So they've just published their phase 1 data on their MERS vaccine using the same vector, and again, disappointing levels of neutralizing antibodies stimulated by that vaccine. Another important issue that needs to be considered with developing a SARS-CoV-2 vaccine, and which has been raised by efforts to develop SARS-CoV-1 and MERS vaccines, is the risk of vaccine-enhanced disease. And this has been reported in some, but not all, animal models. So this is a paper from 2011 looking at an activated SARS-CoV-1 vaccine in mice. And what they showed was that if you vaccinate aged mice with this double inactivated SARS vaccine, with or without alum as an adjuvant, that you get partial protection, whereas if you vaccinate young mice, you get good protection. So looking here at the antibody responses, so you get enhanced antibody response if you use alum, if you don't, and that's against 2 different targets. And it also provides protection. This looks like it's moved around slightly. Apologies for that. But this has also been cropped. But there's some eosinophilic infiltrate, while it also protected partially against disease. And sorry, those graphs have been cropped by from going from a Mac to a PC. While it also protected against disease partially and better with the alum, there was still an infiltrate seen in the lungs of the mice that had been vaccinated with the inactivated vaccine, whether with alum or with not, and there was infiltrate of eosinophils into the lungs. Oh, sorry, yep, here it is up here. So the eosinophils are seen in the lungs, and we can count the eosinophils, and we can see that even with the inactivated vaccine on its own, there's more eosinophils and more with the alum, enhanced with the alum, suggesting an allergic type response being stimulated by the vaccine. And indeed, if they looked at expression of cytokines that are associated with that Th2 allergic response, they saw enhanced IL-5 and IL-13 and CCL11, which is a chemotaxin that brings in, or chemokine that recruits eosinophils into the site of infection, was seen post-infection in animals who had been vaccinated with this vaccine. Another form of immunopathology has been reported in non-human primates. This seems to be more antibody dependent rather than mediated by Th2 immune responses. So this was reported last year in monkeys. So these are Chinese rhesus macaques that were vaccinated with an MVA, so a viral vector expressing the spike protein or a control protein. And then they were challenged 4 weeks after the boost with SARS coronavirus. And the vaccine stimulated really good antibody responses that seem to persist. and these are the control animals who developed an antibody response after they were infected, and those, it seemed to provide protection against viral replication. So in the blue, we can see that this is in the nasopharynx, suppression of viral infection, but there was worse pathological changes in the lungs in the monkeys that had received the vaccine prior to infection, looking at days 7 and days 35 after infection. And if they gave antibody instead, if they gave antibody alone instead of vaccine a couple of days before infection, they could replicate those changes in the lungs. And it seemed to be due to macrophage activation. So these are in the control monkeys. So the macrophages in the lungs were lesser in number and were mainly producing the anti-inflammatory cytokine TGF-beta, whereas in those that had received the antibody prior to infection, they had more macrophages and mainly produced interleukin-6, which is a pro-inflammatory cytokine. So this has been recognised by a number of vaccine developers led by CEPI, and there's concern about this with SARS-CoV-2 vaccines, and they've come up with consensus considerations that need to be taken into account for vaccine development. Nonetheless, a vaccine is desperately required, and taking these all these things into consideration and the severity of infection globally at the moment, there's a strong desire for rapid deployment of vaccines. Normally, vaccine development takes a linear process where you've got preclinical testing and a variety of animal models that might take 5 to 10 years, and then through a series of clinical trials testing safety and then immunogenicity and then protection against infection in increasing larger groups, which can take a number of years. And normally it might take 15 years to get a vaccine to market. However, there's strong desire to deliver a vaccine more quickly, and with the experience with what happened with vaccine development with Ebola, we've got a compressed timeline now. So we're looking, the approach that's being taken at the moment is that for many vaccines, phase 1 trials are being conducted simultaneously with this preclinical testing, and then we're seeing simultaneous conduct of phase 2 and then phase 3 trials to try and deliver a vaccine candidate within the sort of 18-month time frame or less. It should be made, the point should be made at this point that the quickest vaccine development process in the past was 4 years for a mumps vaccine. So if we manage to pull this off as a global research community, this will be an outstanding effort. There are a number of vaccines that are vaccine programs that are in process, and a number that are in clinical development, and we've already got 11 phase 1 trials, 8 phase 2 and 3 phase 3 trials, and these involve a number of different technologies, and I'll just sort of take you through some of the highlights of that now. So the first is inactivated viral vaccines, and there's 3 groups in China that are pursuing this. And the first publication was in Science, and this is again Sinovac, who had worked on the SARS-CoV-1, and they showed that using an alum adjuvant in 2 doses, they were able to stimulate good levels of neutralizing antibodies. So by 6 weeks, they're up to sort of 1,000, an EC50 of about 1,000, which is higher than that's seen in humans who've recovered from SARS-CoV-2 infection. And if you vaccinate monkeys and then challenge them with the virus, there's good protection with the higher dose against both infection in the nasopharynx and throat and in the lungs against infection. And there's no reports of immunopathology, which is good, and that was with 3 doses. They haven't published their phase 1 or phase 2 data yet, but there's a news report out saying that that it looks promising, so no severe adverse events and they were able to stimulate neutralising antibodies in over 90% of patients or volunteers, and they're now planning phase 3 trials in Brazil. There's also Sinopharm, who are working with the Wuhan Institute of Biological Products, who've also got an inactivated viral vaccine. And they haven't published any data yet, but there's again press release suggesting that they're getting very high rates of seroconversion with 2 doses, and they've tried various different durations between the doses. Both of these are not in the highest risk populations, but they are seeing seroconversion. Another type of vaccine that is progressing through clinical trials are viral vectored vaccines. One of these is an adenoviral vector. So this is using another cold virus, and this is by CanSino, which is another Chinese company, and they have conducted they've published their phase 1 trial in the Lancet. And they were able to see neutralising antibodies, although the levels are not particularly high. So they up to sort of 30 to 40 titer. Sorry, here's the neutralising antibodies here, sorry. So yeah, even less than 30. And they also were able to show T-cell responses as well. But of note, these are the people who've got pre-existing immunity to Ad5. So Ad5 has got is a high rate of seropositivity in the community. Roughly 50% of people have pre-existing immunity, which might prevent or hinder the vector from infecting cells. Certainly they saw lower cellular immunity in people who had a pre-existing immunity. Nonetheless, they have, this was in the news a couple of days ago, this has been approved for use in the military. And they're also progressing to phase 3 trials. The other, so trying to get around this issue of pre-existing immunity is using the simian adenoviruses, such as the Oxford group that's using a chimp adeno, so similar to what they used in their MERS vaccine, and they've published their monkey data, and they showed that it stimulated neutralising antibodies, and it prevented infection in the lungs, but it didn't have any effect on infection in the nasopharynx, and they saw protection against pneumonia, against severe pneumonia in the monkeys who had been challenged with SARS-CoV-2. So this was perhaps a slightly disappointing result. They have completed or in the process of completing their phase 1/2 trial. There's been no data released yet, and they're planning phase 2/3 trial in the UK and a couple of phase 3 sites in Brazil and South Africa. And of note, there's a preprint that's just been released that suggests that maybe you need a second dose of this vaccine to get much better levels of antibodies. So they boosted mice and pigs, and in both they saw if they gave a second dose, they saw much better antibody responses. There's also a DNA vaccine candidate from Inovio that is progressing. So again, they've shown neutralising antibody responses, not at massively high levels on their preclinical data, and also been able to stimulate good T-cell responses as well. There's a phase 1 trial results have been announced but not published, so not available for analysis yet, but they say that they've got immune responses in most of their recipients, although they don't specify humoral versus cellular, and they say that it provided protection in a preclinical challenge model, and that non-human primate studies are underway, but none of that data is available at the moment. The other vaccine candidate that has stimulated great interest are the RNA vaccines. So Moderna. So there's been no RNA vaccines that ever made it to the clinic yet, but there's suggestion that these could be promising for the future. So these are like DNA vaccines, but they're RNA encoding the gene of interest, usually the spike, and they encapsulate it in a lipid nanoparticle to deliver that to the cells. And Moderna progressed to phase 1 trials in parallel with their preclinical animal data, and they announced that in preliminary data, they saw good neutralizing antibody levels in 8 of their 155 participants, and they got a massive investment on the stock market in response to that. They've got phase 2 trials underway. They've just published some preclinical data, and it does seem to produce quite good neutralizing antibody responses. But it varies between different mouse strains, and it did provide protection as well, depending on the amount that you gave. And they importantly saw no enhancement of disease if you had a suboptimal antibody response. And just come out to yesterday, or actually today, Pfizer have got their own RNA vaccine, which they're developing with BioNTech. And again, they've published their phase 1/2 study results, and they've shown good neutralizing antibody responses as well. There's also protein subunit vaccines, and I'm just going to talk about Novavax. Sorry, I realise this is running slightly over time. So they've got a spike glycoprotein that's in a nanoparticle, and they give it with their own proprietary saponin-like adjuvant, matrix M, and they've shown excellent neutralizing antibody responses in a paper that just came out yesterday, in neutralizing antibody responses in mice, and that they were able to provide protection against infection in mice that were challenged, and that they got greater protection with the matrix M adjuvant and with 2 doses, and this protected against disease. So at the top here are the mice that have been had the highest dose of the vaccine, and they're completely protected against disease. And they saw no worsening with incomplete immunity, and they also saw good immunogenicity in baboons as well, a non-human primate model. So that's now progressing. They've already started a phase 1 trial in Australia, and they're assessing with and without adjuvant at a couple of doses. So things are that we've got lots of irons in the fire, as it were. There are lots of vaccines that are potentially being developed, and some showing more promise than others. But that having a successful vaccine in phase 3 clinical trials is only going to be part of the story. What's going to be a massive part of the challenge is going to be manufacturing enough vaccine to vaccinate the world's population. This is going to need standing up of a massive number of facilities to manufacture, and this can't be done in a linear way either. This is being done in parallel with development. So many of these companies are investing in upscaling of their manufacturing capacity prior to clinical trial success, and we're seeing massive investments in trying to deliver billions of doses of vaccine within the next few years. Cold chain is also going to be a massive issue for some of these. So the RNA vaccines need need to be stored at very low temperatures, which is completely different to the vaccines we have at the moment, and is going to challenge our ability to deliver these vaccines. So there's great desire for equitable distribution, and to try and ensure that there's cooperation around vaccine development. And recently the EU hosted talks, and there was pledge of money to CEPI and to Gavi, and to try and ensure equitable distribution of vaccines. However, whether this actually occurs in practice remains to be seen. We've already seen a report of the US government trying to purchase a European vaccine producer, CureVac. There was comments by the Indian chief executive of the Serum Institute of India, who are the largest vaccine producer in the world, suggesting that most of the vaccines would have to go to their own countrymen before it goes abroad. And really the political reality might be that it's going to be very hard for any government to allow vaccine produced in their own country to be exported while there's a major problem at home. And as Miguel mentioned, we've just in the last day or so, we've seen the US government purchase up the entire world stock of remdesivir. So there has to be some concern that this may happen with vaccines as well. The other challenge that will present us once we get it is how we actually deliver it. Who's going to get the first doses? How are we going to store these vaccines throughout the vaccine delivery network? Who's going to administer the vaccine? So these are all issues that are going to challenge governments. And just to finish with, we have seen that the New Zealand government has committed to allocated 37 million to COVID-19 vaccine strategy. So part of this is for New Zealand research and to try and support manufacturing capacity in New Zealand to try and contribute to that global upscale of manufacturing. They've also contributed to CEPI and to Gavi around the equitable distribution, and really important to see that they've made that commitment to equitable distribution, and particularly ensuring our Pacific Island partners can access a vaccine when needed. So apologies for going slightly over. So time for a couple of questions perhaps.
Thanks, James. Perhaps we do, just one or two. Let's see. Another enlightening presentation. Thank you, James. What are the risks, if any, of an expedited vaccine process? Would efficacy be an issue?
Yeah, so I think that it's likely that the first lot of vaccines that become available are not necessarily going to be the best vaccines. They may not be the ones that stimulate the strongest immune response or the most durable immune response, and that they may require boosting down the track. So I think that there's a risk that, that those first vaccines may not be the best, and there's already huge commitment to manufacturing those vaccines. Those projected doses, I should say as well, might come down if we need to give a booster, so you can halve the AstraZeneca dose, number of doses if you need to give a booster of the of that Chimpadno vaccine to stimulate a good immune response. There's also, I guess, increased risk of the vaccine-mediated immune enhancement if that is going to occur. We haven't seen any good evidence that that's going to occur to date with the SARS-CoV-2 vaccines, but there is a risk of missing the less common side effects that might not become evident until vaccines are already starting to be rolled out. So having data, vaccine safety monitoring is going to be really important.
Mm-hmm. What is the limiting factor in seroconversion? Will we need to be continually exposed to the virus, or is there an option for herd immunity in the long run?
So I think part of the problem, it depends on the vaccine platform. So some vaccines are just not as good at stimulating antibodies, such as the DNA vaccines, whereas others are much better. T-cell help is also going to be really important, and I think that's increasingly being recognised. And while a lot of these early vaccines are focusing on the spike protein, there may not be as many T-cell epitopes for providing T-cell help in the spike protein as there are in other parts of the virus, so we may not get such enduring, such strong neutralizing antibody responses. The reality is, especially if we look at some of the data from these early vaccine studies, is that we are likely to still see circulation of the virus in the community, so there probably will be some natural boosting that occurs as well. Hopefully, if people are reinfected and have partial immunity, hopefully they have less severe disease, although we don't know that to date, but the animal studies would suggest at least that partial immunity provides partial protection rather than causing enhanced disease. So it may be that suboptimal first vaccines are still are good enough and help get us out of our current hole while we try and develop better second generation vaccines for SARS-CoV-2.
So research and development underway.
Yeah.
Right, that's all we have time for today. Thanks once again to both our speakers. We will be back for the last instalment of the COVID-19 Masterclass tomorrow at 12. Join us then. Ngā mihi.